Morning and welcome to the first session of Health Matters. My name is Jeff Child. I'm a born and local resident and a member of Stanford's Community Council. It's nice to see Stanford providing this opportunity for so many people to come and learn about healthcare and things that they're interested in. So it's great to see you all here. I would like to thank you and introduce you to Dr. Ewan Ashley. You probably think you know him because you've been looking at his face for weeks now. He is the poster boy for our conference. So he is the Associate Professor of Cardiovascular Medicine, Genetics, and Pathology at Stanford Medicine. And he's going to give us a sneak peek into the genetics of elite athletes. And just so he knows the room, is anyone here an elite athlete? We don't want to offend anyone if he says things about you. So since most of us, or I should say now all of us, are not super athletes, I think it's going to be interesting to find out how really understanding them might help all of us. So without further ado, let me introduce Dr. Ashley.
All right, well, thank you for the great introduction. It's a pleasure to be here. This is, I guess, my home turf, so it's nice to be speaking, and thank you for coming and being part of the community day here. I am excited to tell you about super athletes, superhumans, and what we might be able to learn from them to help us treat patients better. So here's one superhuman we're going to be talking about a little bit later. Feel free to tweet anything you hear here. It's all pretty public. Well, I wanted to introduce you to Emery-Dreyfuss. He's a Finnish cross-country skier. One of the best and most successful cross-country skiers ever in the world. He actually won 7 Olympic medals, several gold, silver, and bronze. And in a country that's marginally obsessed with cross-country skiing, he was one of the best ever. But yet his career was somewhat, had a shadow over it, because every time he had his blood tested, his hemoglobin count was high. So hemoglobin is the molecule that goes into that's in your red blood cells that allows you to carry oxygen. And you may have heard of something called blood doping. Blood doping is something that some of our elite athletes have been found guilty of, and it's illegal. It's not allowed in sport. So why would you blood dope? Well, and what is it? Well, blood doping is when you take your own blood out of the body, you put it in a bag, you put it in the fridge, and you wait till your own body makes up those blood cells, and then just before your race, you give yourself the red blood cells back. They're your red blood cells, so there's no problem, there's no immune reaction to them. And what you end up with is more red blood cells than you had before, more oxygen carrying capacity, and the ability to perform at a higher level. It's illegal. It's not allowed in sport. There are legal ways to make your red blood cell count go higher. You might have heard of altitude training. You can go up to high altitude. The oxygen partial pressure is lower, which means your body feels like a lower oxygen and builds you more red blood cells so that you can use them. And you can even train high, and if you live at the low part, you can train high. You can live at the high part. Some people move to high altitude to try and improve their performance. Athletes will do anything to win. So you can train high, live low. You can live low, train high. You can live high and train even higher than that. This is all to make more red blood cells. And how many red blood cells do you have? You normally have about 40 to 50%. If we take a vial of your blood and spin it down, let it settle, about 40 to 50% is red blood cells. Well, this guy, if you spun his blood down, almost 70% of his blood was red blood cells. That's pretty significant. So you could see that if you were winning a whole bunch of gold medals and you're in a world where blood doping exists, you're going to have a lot of suspicion come your way. But it turned out that Emery-Dreyfuss had never blood doped in his life. He actually had a condition called erythrocytosis. And this is his family tree. And you can see it. In fact, he's right here with the arrow right here. This is Euro. And it turned out that he had a mutation in his EPO receptor. So you might have heard in the modern era, you can increase your red blood cell count not just by blood doping, but by injecting yourself with erythropoietin, which is the hormone that your body uses to tell the bone marrow to make more red blood cells. And so you can do that too. You can inject yourself with EPO. And there are famous athletes such as Lance Armstrong who've been known to do that as well as blood doping. But this was actually genetic doping, and natural genetic doping at that. This receptor in the erythropoietin, the mutation in the erythropoietin receptor led, in Ero's case, to kind of the accelerator being pushed down on his body's propensity to make red blood cells, and he made more and more. In fact, every time you see a dark square or circle here is somebody in Eero's family who carried the same mutation and had a very high blood count. He had several elite athletes in his family who won multiple medals in cross-country skiing, in other words. So he was a superhuman. And the question for the next half hour for us is, can we learn from superhumans like Eero to let us treat people who have, for example, low blood count? Can we learn in a way that helps us help athletes, learn from athletes in a way that lets us help patients? And that's the question that we'll address. Here's him doing his favorite sport. Of course, environment is part of this too. It's not, you can't get to become a gold medalist with just the genetic complement. You can't sit at home and eat Doritos and watch TV and win a gold medal. That doesn't happen. It doesn't matter how good your genetics are, right? And it doesn't matter how much you train. If you don't have the right genetics, you're not going to win a gold medal either. And so he trained a lot. Actually, he would do many, many miles, even as part of his job in delivering to remote areas of Lapland where he lived. Well, here's another story. Some of you might even have heard this story. It was on NPR recently, and part of this ProPublica story. You might even have heard, if you're at this talk, it's possible you've heard of a book called The Sports Gene by David Epstein. And this was a follow-up article by David Epstein as a follow-up to his book. And it starts with this person here, Jill Viles. And Jill Viles had a muscle wasting disease, and she was aware that she was very weak and her muscles were small, and she could see her veins sticking out. And this was something that was clear in her family. She'd spotted this in part of her other family members too. And she would go to doctor after doctor after doctor and tell them, and try to work out what she had. In fact, she started, by the time the internet came along, Googling and working out what she had, and she thought she had something called Emery-Dreyfuss muscular dystrophy. Muscular dystrophy is just a weakening of the heart muscle. There are lots of genetic versions of it, and Emery-Dreyfuss is one where you get particular contractures of the elbows and the heels. She was convinced she had it to the point, if you read this article, that she would reach over and write on her chart, patient has Emery-Dreyfuss muscular dystrophy, even though the doctors were saying, I don't think you have this, and you don't fit, there's no other way. And so she is a citizen patient and a citizen scientist of the highest order. I recommend the same to you, although now you'll have to reach over and type because we don't have, we don't have paper notes anymore. But this is her, look at her very thin muscles. But she also noticed something else, which is in herself and in her sister, that those muscles were very well defined. They were small and wasted, but they were very well defined, and she could see the veins very clearly. And in fact, some of the muscles seemed actually a little bulkier than they should be. And she was aware of this athlete here, and this athlete is Priscilla Lopez-Schleep, who's one of the best sprinters, hurdlers that Canada has ever known, has multiple world and Olympic medals. And she looked at the muscle definition and thought, well, this person has a muscle problem, but has obviously huge muscles and is a world-class sprinter, and I have a muscle problem, and I have very small muscles, and in many cases, she was moving in the last few years towards her last few steps and is now wheelchair-bound. And she came up with this theory that she had that they had the same gene that was malfunctioning. And so she wrote to David Epstein, shown here, and she said, I think we have the same gene. And he received thousands of these letters after publishing the book. And he says in the article, at first, he would throw them away. But then he realized that she'd collected, and this is a page from her notebook, like dozens and dozens, hundreds of pages of sort of citizen science connecting these two things. And she had this theory, because by that point, she had actually been diagnosed with Emery-Dreyfuss. And so she'd been proven right. And a gene called lamin, which is a gene that encodes a protein of the nuclear envelope. So the nucleus, as you remember, holds the DNA in the middle of every cell. And the envelope around it is an important, important kind of container. And actually, the skeleton of that nucleus is very important too. And lamin encodes for the filaments of that skeleton. And so she had this theory that Priscilla Lopez had a same problem that just manifests in a different way. And that's what this pathway is here. That here you can have an anabolic pathway and build up muscles. And here you can start with the same mutation or a different mutation in the same gene and end up with this, a muscular dystrophy. Well, of course, I'm telling you the story because she was absolutely right. She managed through David Epstein to track down this athlete. She met with her. She asked her if she'd be willing to be tested, and she was tested and has a lamin mutation. It's important because they actually share another condition, not just Emery-Dreyfuss muscular dystrophy, but a condition called partial lipodystrophy. And lipodystrophy means that the fat in your body kind of atrophies away. And that's why the muscles are so well defined in both of these people. But the thing about lipodystrophy is you also have very high triglycerides circulating around your blood. And you're at risk for something called pancreatitis. Pancreatitis is inflammation of your pancreas. It's one of the most painful conditions known. It can be life-threatening. And if you don't know you have very high triglycerides, and because you're a young athlete, you've never had your cholesterol measured, so you've never had your triglycerides measured, you wouldn't know that. And so one of the things that happened as a result of them realizing this gene was causing these very different pictures was that they managed to save a potential future pancreatitis from happening to Priscilla. But how can one gene cause all these different conditions? In fact, it's the same gene that causes this condition here, which is called progeria. You might have heard of Benjamin Button disease. Sometimes it's referred to that. Early aging, where young kids start to look as if they're 80 years old. It's the same gene. It's the same gene that causes dilated cardiomyopathy. Your normal heart goes from this size to this size and gets wider, gets weaker, has an inability to pump enough blood to the tissues. So this same gene, in fact, if you look at the top line, you'll see that the lipodystrophy, this is adipose tissue, is fat. So if you mutate the gene over here, you can get that. Right along the gene, you can get this dilated cardiomyopathy. Also, right along the gene in different spots, you can get skeletal muscle. It's just an incredibly complicated gene. You can mutate it in lots of different ways, or it can become mutated in different ways and cause all these different diseases. So we can learn a lot from athletes about lots of different diseases. And so I wanted to tell you a story. Both those stories came from other people, of course. These were historical figures, and in the other case, actually, that story was just out this year, so people undergoing very current challenges. And so I wanted to tell you about this. This was a story we did, at least a piece of science that we did a few years ago now. And I describe it as ultra-endurance lunacy in the Highlands of Scotland. And your first question on seeing this slide is probably, where is Kellycrankie anyway? You probably noticed I don't come from this country, or I have a funny accent. I came from Scotland originally, not from Kellycrankie. I came from a larger town called Glasgow. But Kellycrankie is right there. Here's a picture of Scotland, and it's right in the middle at this point here. And you were thinking, why is Kellycrankie important? Well, if we just start historically for a minute, there was a battle there because Scotland liked to fight other people, usually the English. And of course, when you've had a battle, whether you win or lose, you write songs. Usually you write songs that are very maudlin and sad, but here, and Robert Burns is our most famous poet, and he's famous for writing Auld Lang Syne and other tunes you might have heard of. My love is like a red, red rose. I will not sing for you. That would be important. That would be important for you to know. But he did write a song called The Battle of Kellycrankie, which has a verse, and ye had been where I hae been, ye hadna been sae canty o, and ye had seen what I hae seen in the braes of Kellycrankie o. So you imagine a man with a beard and a folk song singing that. Anyway, that's Kellycrankie. It's important because, and you're wondering why I'm telling you this, aren't you? It's important because, but before I answer the question, actually, I'll tell you why it's important. Here's another question that I'm going to answer first. And then it will all become clear. Believe me, it'll all become clear. What do super athletes have to do with the rise of reality television? Anyone have any idea? Anyone heard of this guy? Ever heard of Mark Burnett? It's not Trump, yeah. I was trying to get through the whole morning without mentioning Donald Trump. But so Mark Burnett is an interesting guy. He's British, actually, originally. And he joined the British Army, I think in the 80s. And he went into the parachute regiment. The parachute regiment's a bit like the Navy SEALs. It's like the hardcore part of the British Army. And he left, and then he moved to LA and was just doing various jobs. And he got interested in doing a race. At the time, something called adventure racing had kind of become invented. A guy called Gérard Foussille, a French guy, had come up with an idea to do something called an adventure race. And he built something called the Raid Gauloise, a French adventure race where people would come and they would test themselves to the limit, essentially. What they would do is they would do hiking, they do trekking, they do mountaineering, they do biking, they do kayaking, they might do even horse riding at times. Basically, he would take them into the wild, and for days on end, he would test them to their limit. And you would compete generally in teams. And this became very well known and very famous at the time in the early 1990s. And Mark Burnett actually put a team together and joined this and realized that this was kind of an amazing thing. And so he bought the rights for another adventure race called Eco Challenge and turned it into a TV show. And you might have heard of the TV show. It became essentially what was Survivor. And Survivor basically kicked off, by popular opinion, the whole slew of reality television. So Are You Smarter Than a Fifth Grader? The Real Housewives of Beverly Hills. You can blame Mark Burnett for that, okay? And in fact, you could blame adventure racing because he got into TV. He's now like a president of a TV company. You could blame him for that, and he got into it because of adventure racing. And so I'm going to tell you about another adventure race. This one happens in Scotland and around Kellycrankie, and that's why I mentioned Kellycrankie a moment ago. But before we get to show you the athletes who were participating and telling you just what they did, I want to stop for a minute and just think about what happens inside your body when you start to exercise. Because these guys are pushing their bodies to the limit. First of all, when you start to exercise, the physiological changes that happen actually happen even before you start. So the minute you start to think that you're going to exercise, right, you're pulling on your sneakers, you're thinking about going to the basketball gym, whatever, as you get started, before you even start running, your body is already switching you into the mode of getting you ready to do exercise. Your heart rate is rising a little bit. The blood is starting to be shunted to the muscles that are going to need it. Your sympathetic or fight or flight system is starting to widen your eyes so that you can see more clearly. It's many things start to happen. Your rest and relaxation, internal nervous system is starting to shut down the blood flow to your gut because you don't need to be digesting food. You've got to be ready to run. And this all happens before you even start. And when you start exercising, a number of things start happening. Of course, your heart rate rises, but you don't get much more blood flow if all that happens is your heart goes faster, because what happens is your muscles start to move, and they start to pump blood back to your heart. And your heart is the single most amazing organ in your body. I'm biased, I'm a cardiologist, but I believe that, right? One of the things that makes it really amazing is you pump more blood into the heart, it pumps more blood out. Think about that for a minute. There's no reason why it should have to be the case. But something called the Frank-Starling mechanism, and that's a mechanism that, actually there were two people, Frank and Starling. A lot of people think they're one person called Frank Starling, they're not, it's two people. They never met. But they described a law that said if you pump more blood into the heart, it responds with a more forceful contraction, pushes more blood out. It's why, and many of you are young in the room, and if you're not young, you're young at heart, but it's why I could take your heart rates and drop your heart rates from 60, 70, that they're sitting relaxed, hopefully listening to this, and make them 20, and you probably wouldn't even feel it if you have a young and compliant heart. Let's say you have a diseased and older heart, and you have a heart rate of 80 beats per minute, and I drop it to 20, you'll probably immediately pass out, because your heart isn't capable of the same Frank-Starling mechanism, it's not as compliant, it doesn't stretch as well. And that's why when you get rhythms like atrial fibrillation, which is the top chambers of the heart wobbling a bit too much and your heart rate goes too fast, if you're young, you might not even notice it. If your heart is stiffer and you're older, you probably will get symptoms and you might well feel lightheaded and dizzy. But that mechanism is exactly what allows athletes to increase their cardiac output. So your heart rate goes up, but the amount of blood per beat that your heart pushes out goes up too, and you get this huge increase in your cardiac output. And if you're an elite athlete, you have an absolutely massive increase in your cardiac output. Your heart isn't much bigger, the walls are not much bigger, so if you read about athletes having massive hearts, it's actually really not true. It's very, very minor changes, but important changes. If anything, they're way more compliant, and that's key. And so that's how you can sustain this exercise, and you can sustain a particular level of exercise almost indefinitely until your fuel supplies run out, and you change your fuel as you go. And that's important for certain events, like the one I'm going to tell you about, which is called adrenaline rush. This is the cardiac output from the different parts I mentioned. It's a little bit grainy, but you can see at the beginning the blood flow, which is 5 liters down here at the beginning, and is going 20% to the muscles, 4% here to the fat, 15% to your brain, and 25% to your gut. When you start exercising, look at the difference. Your gut shuts down 3 to 5%. Most of it goes to your exercising muscles. And that starts before you even begin to exercise and can continue almost indefinitely. Here's a zoomed in map of Scotland. So this adrenaline rush, as it was called, was a very interesting adventure race. So adventure race, you had to compete in teams. One member has to be of the opposite sex, so it's co-ed teams. And this was a continuous event. So some of these adventure races were run with enforced sleep periods. If you're in a dangerous part of the world, they say you shouldn't be out at night. When it's dark, you're going to rest. We're harder than that in Scotland. I just want to tell you that, right? You don't get any rest. There are no enforced sleep periods. If you want to keep going permanently through this whole race, you can do it. And the winning team that I'll show you in a minute, they did that. They slept essentially not at all. And sleep deprivation becomes a huge part of the physiology of these athletes. So this is a zoomed in picture of Scotland. You can see some of the things that we're doing. Cycle here, trek over here, and there's various different other things. It's a little grainy, so you can't read it all. So here is the heart rate tracing from the woman who, so the team that won was 3 men and 1 woman. The woman actually went to join the army after this was finished, the Marine Corps. And this is the heart rate tracing from Polar Heart Rate Monitors a few years ago. And this is through the race duration. I haven't told you how long it was. I've told you they don't sleep. It was 5 days long. So her resting heart rate was 40 beats per minute, actually even a little lower, 30 something. And look at where it's at. It's at 140 for 92 hours. And they had approximately, they took turns to try and sleep for 10 or 20 minutes during this time, but they basically never slept. At one point, they were in a kayak, all 4 of them in a kayak, and they were taking turns to sleep. Like they take 10 minutes each while 3 of them moved. And at one point, all 4 of them fell asleep and they fell in. This is the sort of thing that happens during this race. So it's really pushing people to the absolute limit and to the extremes. You'll see some pictures in the next few slides from the actual event. So one of the things that happens when you exercise that long and that hard is yeah, I'll do questions at the end if that's okay. Is one of the things that happens is your heart actually gets weaker. Not by much, and not into the range of someone whose heart is failing, but it gets a little bit weaker. And so our study was actually the one at this point to show the longest and most severe form of this weakness. It's still not causing heart failure by any means, but the hearts of these athletes actually tire a little bit, something called cardiac fatigue. And so these are the various studies with the length of race along here and the drop in something called fractional shortening. It's the amount that your heart contracts per beat. And you can see ours is up here. So still not into the range that people are having heart failure and being admitted to hospital, although a few people are admitted to hospital. It's not for heart failure. It's usually for environmental stress, as you could see. But it's pretty crazy what happens. I mentioned the internal nervous system. How do you get a sense of the internal nervous system, short of sticking probes in nerves, which we didn't really want to do? You can actually look at the frequency of changes in some very easily measured parameters, heart rate and blood pressure, if you can measure them continuously. So the way to think about this, you can break down a signal. For those of you who are more technical, it's called Fourier analysis, but for those of you who are not, think of it like this. If I wanted to study a population of people over time, you might follow them for weeks or months or years. You would see some things that recur on a weekly basis. You know, every Monday this happens, and you'd see a frequency of that. There were some things that might happen on a monthly basis, and then there's some things that would only happen once a year. So you can break down the signals of looking at a population of people over the course of the year into these different frequencies. Well, you can do that with the rate, the distance between the heart rates and blood pressures. And that's what is shown here, heart rate at the top and blood pressure at the bottom. And we break this into a very low frequency, a high frequency component that give us a snapshot, a window into that internal nervous system. And you look at the change before and after. And this is just one athlete. This is a few minutes over which we look. And you can see the shape of the peak here. This very low frequency component is higher after. And in the blood pressure, these little peaks, it's hard to tell, but these are higher here, and this is lower afterwards. So we get a sense that the people who are spending 100 hours with their fight or flight response turned absolutely full on get a slightly reduced function afterwards and have this is indicating thermoregulatory changes, which is not surprising when you think of the race, and then also changes in this internal nervous system, the fight and flight and balance versus the rest and relaxation. One of the other things we do then is look, this is in the early days. I'm going to tell you in a minute about what we do for gene testing today. This was many years ago when we were lucky if we could look at one gene. So we did. We looked at one gene. And it was the only gene that at that point had been associated with high performance. And it's the same gene, if any of you are on medication here for blood pressure, you might have heard of, if you're not, a medication called an ACE inhibitor. Stands for angiotensin converting enzyme. And in fact, that same gene, angiotensin converting enzyme in the body, is responsible and contributory towards performance. In fact, when we split the athletes by how much their heart reduced in function, by their particular type of this ACE gene that they had, we found that it actually differentiated them quite well. And so this is one of my earliest indications, anyway, in the work that we did, that the genes might be important and might give us some kind of insight into what we might want to do in the future with this kind of work. Well, the work, actually, when you tried to put this together, there was no funding for this. We went into the Scottish Highlands. We had some portable equipment. I persuaded people to lend us it because we didn't own any of it because I had no money because I was a PhD student at the time. And as you'll see from my acknowledgements here, I also got my father and my wife to help, which is about the only way that's done. But also it became something that became featured on the BBC Science programme, so I thought you might like to see a little video of it.
Everyone exhausted. His team are waiting for a
They looked at two different teams.
They grabbed 10 minutes sleep, at first in nearly 2 days.
Attractive Scottish Highlands.
But we've got a nasty surprise in store for them. The ferry stopped short of the shore. Wet and cold, they head into the night. After 37 hours non-stop, their bodies are flooded with adrenaline. Their heart beats twice as fast as normal and untiring.
It's about right, broken bodies.
The weather has seriously deteriorated. It's getting dangerous. 77 hours into the race, Caroline's team have only had 5 hours sleep, and they're beginning to feel the strain.
So the winning team, as I mentioned, no, listen.
We start seeing things that might coming through to the shadows, ticking like 2 dancing bears in front of me. It looked like a dinosaur, I wasn't quite sure. Triceratops, which turned into a Highland cow, which struck. These a cup of tea. And it's also signals you are getting very fatigued.
So sleep deprivation is a huge part of this. In fact, although we were focused on the cardiovascular physiology, we were quite interested in the psyche of these people, first of all, for doing the race in the first place. But secondly, 90, 100 hours with minimal sleep. This team slept about five or 7 hours, and the whole time, as I mentioned, the winning team basically had no sleep rest in the whole time.
Finally, after 113 hours on the go, Caroline's team are on the home straight. They come in.
It was actually a science program, so you'll see like 10 seconds of science now.
He has examined some of the finishers' blood. He was right. A surprising number of them do have a difference in their genes for fitness. So perhaps they were born.
There's a young guy coming up now that you'll see.
But as his team examine them, they find an even more striking way to predict success.
The ones that we found
Who's that guy?
top half of the list were actually, they did actually have different hearts. They were thicker hearts, stronger hearts. That seems to be at least part of the reason that these guys are near the top of the list.
They've covered 400 kilometers, kayaked over 3 lots, cycled for 17 hours, and climbed a mountain. Only 14 teams have made it this far. Caroline's team has succeeded.
So you can see I used to have a Scottish accent, but changed a little now. But you could get a sense of this race is completely bonkers. I mean, it's completely crazy. So the question, though, is can we learn from these people? And so that's what I've been teasing you with a little bit through. Can we learn from people like that to discover new therapies? Do we have reason to believe that by studying the extremes, we're not just having fun or putting ourselves in the wilds in that those kind of conditions to try and learn more about the human condition, which is worth it by itself? But as a cardiologist, can I find a new therapy? Well, there's actually very good evidence just in this last couple of years that by studying the people at the extremes, we can find a new therapy. And to tell you that story, I have to introduce you to cholesterol. Many of you are familiar with cholesterol. But this is a figure essentially showing what happens with cholesterol in the bloodstream. The bloodstream is here at the top, and these yellow round objects are your bad cholesterol, LDL, low-density cholesterol. And there's a receptor on the surface that grabs the low-density cholesterol and pulls it into the cell. But there's another mutation in another gene that has this big long name, proprotein convertase subtilisin kexin type 9 serine protease. This has got another 2 names I didn't even include. So you'll be happy that we're going to call it PCSK9 from now on. And this came to light because of an extreme individual. In fact, a series of extreme individuals. They were discovered in Texas. Even before that, actually, there was a group and a couple of families in France who were noted to have mutations in this receptor, and they had particularly high cholesterol, really high cholesterol. They had something called familial hypercholesterolemia. And so this gene had been found, PCSK9, to be a cause of familial hypercholesterolemia. But in this group in Texas, what they found was that there were people with incredibly low cholesterol. In fact, there was an aerobics instructor who became part of the story as this came out, who had a low-density cholesterol, and some of you will be familiar with the numbers. You might want to keep yours below 100 if you had a heart condition. You might want to keep it even below 70. There's plenty of people who have theirs in the 150, 170. If you have it up that range, we probably will get it down. Her resting, no treatment, low-density cholesterol was 14. 14, 1-4. So she had been born with a condition that made her cholesterol extremely low. And she had no disease. She was actually an aerobics instructor. And so some smart people in Texas thought, well, they should study that a bit more and look and try and find out what it was about them that made the cholesterol low. So you can think of PCSK9 like kind of the controller of a trash collection agency, right? So this person who sends out the trash collectors every morning, right? And in this case, the trash collectors are these LDL receptors. Now, if you were to sort of shoot some sedative into that trash collector and just make him asleep at the wheel, he's going to forget to call them all back in, right? So they're just going to keep going around the body, cleaning up trash the whole time. And that's basically what happens in her situation. The PCSK9 controls the recycling of this receptor. It's not recycled, so it sits on the surface and it just keeps pulling this bad cholesterol away and metabolizing it. And so we actually published this in a paper, and we'll get to that. We published this in a paper just a few years ago and looked at this population. And you can see here on the graph, this is cholesterol up. Here's 150, which is the sort of mean for the population, the average for the population. Here's the people who had these particular changes in their PCSK9 gene. Their average cholesterol is much lower in this kind of range. I mean, hers is obviously way down here. But even more than that, what was really important is it wasn't just changing cholesterol, because sometimes you can change cholesterol and not change outcome, not change heart disease. So here's a picture of, do you have heart disease? Yes. Do you have heart disease? No. Coronary artery disease here. Do you have the gene mutation? Yes. No. So if you have the gene mutation, not only is your cholesterol much lower, but your risk of heart disease is much lower. And that was astounding, especially when you have these superhumans walking around who have these incredibly low cholesterols. So the idea that you could connect gene mutations, if you like, with these superhumans, not just to cholesterol, but to outcome, was really a very tempting thing for the pharmaceutical companies. And the top of this slide shows you that in a very rapid period of time, maybe even the fastest that the drugs have ever been developed and then approved, these PCSK9 inhibitors were approved. And this just happened in the middle of last year. So we moved from a finding to a drug in an incredibly short period of time. We worry that it takes billions of dollars to do that, and it takes 10 years. This happened much faster than that and for much less money. And so I think that my argument to you is that by studying the extremes of the distribution, whether it's athletes or cholesterol or height, we can find new findings, new genes, superhumans, if you like, who are out there, and then we can make drugs to mimic their sort of superpowers to help everyone else. So it can be done. But I was going to tell you that's what leads to our elite study. So you were asked at the beginning if any of you were elite. I noticed nobody put up your hand. Oh, we have an elite athlete now. Good. I'm glad we have some. So we'll see if you're elite enough to be in our study. So if you want to join, you can come to elite.stanford.edu. But let me tell you about the study first before you decide if you want to participate. Now, one of the reasons, of course, that we think athletes are a particularly good model for this, looking at the extremes, is that, as I mentioned earlier, to win a gold medal, you need the genetic complement to be right, and you need the environmental training. But you need the genetic complement to be right everywhere. It can't just be that you have a great heart function, because that won't get you there. It can't be that you just have great lung function, or even that your heart and lungs are great. You actually need all of these. You need to be able to transport oxygen well. That's the red blood cells we talked about at the beginning. You need to have great skeletal muscles to move you fast along short or long distances. And you need to have a great oxygen delivery system. And if all of those aren't in absolute optimum condition, you're not going to be winning a gold medal. So how do we measure fitness? We measure it using a test called VO2 Max, or cardiopulmonary test. Has anyone had one of those? You run on a treadmill, you put a mask on, and it measures the oxygen that you extract from the air. So it's not immediately obvious why that should be the measure that we use for maximum fitness. But in fact, if you think about it, and you're trying to sum all the things I saw in the last slide, your ability of skeletal muscles to demand oxygen, your ability of your body to deliver oxygen, then the oxygen you extract from the air is kind of the sum of all those things. And so VO2 max is something that has been traditionally used now for decades to measure the fitness of individuals and athletes. And normally, most people's VO2 max, is around here. Here's the curve for the population, 35 or 40. It's a bit higher if you're younger. It's a bit lower if you're older. But the average for the population is in here. Well, for this study, we're looking for the people in the 0.00172% of the population over here. But the entry criteria for our study mean that there are people with Olympic gold medals who don't qualify. One of them is this guy. This guy has 7 gold medals, Ryan Lochte. You heard of him? Yeah. His VO2 max is not high enough to get into our study. He's a pretty accomplished guy. He's a pretty good athlete. But it turns out he gets there not through maximizing his VO2 max, although it's very high, and I'm certain it's likely higher than most of the people in the room. It's certainly way higher than mine. So there's no question that that's high, but it's not as high as some of the people like this guy. This guy has the highest VO2 max ever, close to ever recorded. He's in our study, so it's the highest one in our study. There is someone else who has a rumored to have a higher number, but we can't actually verify that for sure. He's Per Olofsson, and you'll notice that he's biting a gold medal, and has some skis here because he's a cross-country skier, and I'll show you why that's come up twice already in a moment. His VO2 max is 92, so one of the highest ever recorded, and he has won multiple world championships and Olympic medals. This is one of the things that if you're an athlete that comes up a lot. It's like athletes are all about competition. And so if you start testing them in the lab, they want to know, did they do better than the last guy, or did they do better than the next one, or did they do better than you? They're all about competition. And so they're also interested in who's the fittest of them all. Like, so which sport? Well, it's not a coincidence that you've seen 2 cross-country skiers. Cross-country skiers have the highest VO2 maxes of any sport. Rowers come pretty close next. Actually, they're down here. Usually we think of them, their ventilator capacity is actually often second, but on this graph anyway, their VO2 max is a little bit lower. So runners up there, cycling, orienteering is kind of like that adventure racing that we just saw a moment ago. So very high. And the thought is that, again, oxygen extraction depends on how many skeletal muscles you're innervating at any one time. And one of the things about cross-country skiing is you're really using almost every muscle in your body, as opposed to cycling, for example, where much of the upper half, although participating in ventilation, is not doing so much, unlike cross-country skiing, where you're using all the muscles in your body. All right, so I talked about measuring genes, and for that other study, we measured one gene. Actually, we only measured one position in one gene. This is one of your genes. I'm not going to show you a whole genome. In fact, if I was to show you a screen like this every second, it would take you 3 months of sitting in this room before you saw the whole genome. So that's a lot. And if you wanted to get a whole genome, even just 10 years ago, you would have needed this much money. $3 billion is what the first human genome cost. It was the Human Genome Project. It took a decade. It took 10 countries. It took thousands of people, and it took $3 billion. Now, if you wanted to sequence a whole genome, it would be significantly cheaper than that. You can do it for $1,000. That's a million-fold reduction in cost. That's like ridiculous. That's like beyond anything we've ever seen. You know, and usually in these kind of forums, we like to do this in Silicon Valley units. So Silicon Valley units are, you know, Ferraris. Although I went past this garage on El Camino yesterday, and it's closed. The Ferrari guy. So I used to drive past every day and see these Ferraris and want one. But on my meager Stanford salary, you know, I can't afford a Ferrari. Anyway, if this Ferrari had dropped in cost as much as human genome sequencing had dropped in cost, you know how much it would cost? It would cost 10 cents. 10 cents. It's a Ferrari for everyone in the room. And certainly, that's one for me as well. So it's a ridiculous thing. I mean, it's an amazing leap forward in technology. And it's been driven by a lot by the area that we live, right around here. Many of the technology, and certainly the bioinformatics that supports it, has come from right around the corner. Well, here's the study, the elite study that I talked about. This is an indication of how interested people are in elite athletes. Is we haven't actually released any of the data yet. I'm going to show you a little bit today. And yet there's already news stories about this study, just the fact that we're doing it. And so this is Mikhail Matson. Actually, he recently qualified for the World Championships in adventure racing. He's in my team here at Stanford, but he is going to be doing, he just came back from Belize a few weeks ago in one of the qualifying championships. So he does that stuff for fun. And we studies it. So this is one of a national class athlete that we're studying as part of this. And this is the lab at Stanford where we do this work. We also test wearable monitors in not just elite athletes, but sort of normal people as well. So the elite study is currently in 11 countries. There are 19 different sites. If you want to get the fittest of the fit, the people at the absolute extremes, the 0.000172% of the population, you've got to go look in all corners of the globe. And so that's what we're doing. You'll see we have quite a few from Sweden because of the cross-country skiing part. We've got quite a few from Europe. In the second phase, we're gonna hit Africa, and we can talk about that later. So far, we've decided to do the genetic studies on mostly individuals of European descent, but it's only because that's phase 1. Phase 2 will be incredibly important to get into Africa. And here's a couple of the results I can share with you. As I said, this work is really ongoing and is just kind of getting started. But this is one of the elite cyclists. And he shares something with, I'm not certain about this person, but a person from this area in Ecuador. And this is someone from the Quechua tribe in the Andes, in the Andean highland region of Ecuador. There are 3 areas in the world where people live at very high altitude. Remember we talked at the beginning about living high and training higher. Well, the Tibetans, the Ethiopian plain, and the Andes are places where people live above 12, 13,000 feet. You'd think that would be a quite interesting place to go to discover extremes. And one of the things that's different between those 3 different populations in the world is that in the Andes, there are 2 groups of people. There are groups of people who have adapted extremely well to living at high altitude. And that's one tribe. And then there's another tribe, of which this individual is one, who have not adapted so well. And there are individuals who get chronic mountain sickness. So if you go up too fast to altitude, you'll start to get short of breath and your lungs will fill with fluid. And that happens to quite a lot of people. If you go too fast, it'll happen to almost everyone. That's why if you go really high, you take your time when you're going up. But there are people who live at that altitude who do not adapt in the same way. And so a very interesting study was done in the past, not by us, but that essentially compared those who adapted with those who didn't and found a number of gene adaptations in those who adapted well. Well, what we're doing is finding that the same gene that they found, it's actually a gene that regulates both oxygen, reactive oxygen species, and thyroid, your thyroid gland, is actually present also in the athletes. So we're trying to tease out exactly how that happens, how that works. But the fact that we're seeing these differences, I mean, it's not surprising that altitude, delivery of oxygen would be important. So we're looking at genes that have significant impact on those functions, and we're finding them in specific elite athletes. We want to find the next Mantaranta, and maybe even find some other things that people share. We can look as well as these individual positions, like the ones I showed you a minute ago. We can look at big changes in genes, and this particular graph shows you big changes, deletions and duplications in the genome. So instead of just one letter being swapped for another, like a big new piece being put in or a big piece being taken out. And we can look specifically at the mitochondria. Mitochondria is the powerhouse of the cell. It's where the energy actually comes from. And this is from a heart, and the heart is packed full of mitochondria. But if we look at the genes that are encoded in there, so these are the ones that are particularly important for metabolism, we also find some signal. And in particular, this is a pretty interesting looking gene here, because in multiple individuals, there are either deletions or duplications. And so we're very interested in following this up and trying to work out both at a cellular level, even a mitochondrial level, by isolating mitochondria, what these kind of changes in these genes do, and then take it all the way through to the athletes to try and help explain why they are world class, but then to take that to patients. For people who have mitochondrial disorders, I see people in my clinic with problems in mitochondrial disorders. We try to work out what's wrong with them. We try to look at all the genes we know. We don't know all the genes. We don't have therapies. We want to make these discoveries to try to connect the fundamental biology of these athletes with patients and then ultimately to therapy. All right, I was going to leave you with some traditional Scottish athletic events. You've seen one already, which is the adventure racing. We do a little bit of tossing the caber, it's called, throwing like large telephone poles. That's not a real sport. I'm just messing with you. And that's not a real sport either, which is drinking single malt, but it's something I like to do on a Friday. I'm happy to take any questions. Thanks for being part of it today. We have a microphone, so if you want to stick your hand up, and then we will bring the mic to you.
Thank you. Dr. Ashby, regarding your comment about studying or understanding the
Mm-hmm.
any relationship.
Well, yeah, so that was what we were thinking about with these people. I mean, maybe even a resistance to sleep deprivation was another. But, and certainly in the course of this documentary that they made for the science program, they interviewed a lot of different athletes, and that clearly was a theme that came through. The basic studying we did was not specifically about thrill seeking, but it's hard to believe that that wouldn't be a highly prominent among these people. When you look at the sorts of things they do, this kind of rope work that you saw on the video is a common part of it. So being at elevation, my colleague who is doing the elite study, who just came back from Belize, they were talking about basically cutting their way through a rainforest, and they're having been told by the locals just to watch out for the snakes. And the snakes were not on the ground, but rather just on the trees. And so they were sort of cutting their way through, and these snakes were like crazy. So it's really quite extreme. I mean, you can see what appealed to Mark Burnett when he decided to turn this into a TV show. So, yeah.
You mentioned how some people
Yeah. Mm-hmm.
studying this affected.
Yeah, absolutely. And I think it's a very complicated position, because it's one that's been embraced to date, mostly around the gender issue, actually, because of there's been some very prominent, well, in fact, this particular sprinter that I mentioned was actually, there was a lot of question about whether she was doping, or maybe whether there was some gender uncertainty there, let's put it. So it's something that sport and exercise and the International Olympic Committee, which is the sort of overall body that regulates the Olympics, it's something that they've embraced before. But what we can do now is embrace, what we have to do and forced to do is embrace it at a much deeper level. Because now we can understand the genetic makeup. As I mentioned, for $1,000, you can have your whole genome sequenced. Now, most of us will not have anything particularly exciting in our genomes, thankfully, right? But the older you are without any significant major medical malady, the more likely it is your genome is not that exciting. But if we take a whole population, and particularly, or if we focus on a group of people who have extreme presentations of disease, for example, when they're kids, we will much more likely find genetic abnormalities. People have asked me, I think they did a recent, this thing was on Sports Illustrated recently, they did a recent feature, and there's an interview, you can find it online somewhere, and they desperately wanted to ask me about gene editing, which might be in your head because it's been in the news a lot, the idea that we now have the capacity to take the machinery that has come from bacteria and essentially do editing of the genome and take out a variant and put in another one. And you could clearly find variants that would be performance enhancing. So that, as anyone, they've asked me, is anyone going to do that? Surely people are going to do that. And certainly athletes would do almost anything to win. I mean, cheat, sure, but almost anything to win. So it was kind of a wacky question. On the other hand, I know a lot of athletes. Fortunately, very few of them cheat. But the reality in the world is that there are plenty of people out there who do, and they're going to look for every opportunity to do that. I think that the key, maybe the best answer to your question is back to the idea of you don't get the gold medal without trying. You can't sit at home and watch TV. Doesn't matter how good your genome is. Doesn't matter how much you inject yourself with red blood cells. You can't jump up and win the marathon. So even people, and in fact, interestingly, Mantaranta, who I mentioned, this guy, he actually never really believed that it was his gene variant that made him succeed. And you could see why he might say that. He felt in some way that was taking away from the humongous effort that it took to train for hours and hours on end for years and years. And he didn't want to just have people think that he was just born that way. But in reality, we're all born, all of the people who win are born that way to an extent, but they also have to train as well. It's a great question. Yeah. Yeah. Thank you. Yeah. Right. No, it's exactly. So it's something we're interested in. I mean, I think because my focus is on cardiovascular physiology, we've made this our primary focus. But some of you, if you read the New York Times, will have seen over the last week. In fact, I think the second part of the story was published today about a friend of mine called Yannis Pitsaladis, a Greek guy, talking about trying to get an athlete to do a sub-2-hour marathon. If you haven't read, there's 2 long-form pieces. I think the second one was this morning. So he actually has a group of us now that we gathered in. It's not hard when he invites you to a Greek island to go and have a conference, right? So we went last year to Santorini and gathered really all the people around the world who are looking at elite athletes, and so half the group were looking at power and performance. Obviously, we were a bit more on the side of aerobic performance, but there's a lot of interest in doing exactly that too. And the interesting thing is, I mentioned we haven't got to Africa yet, but Eastern Africa, especially around the Ethiopian plain and around Kenya, is where a lot of the middle distance gods come from. The people who've been winning all the middle and long distance events. When you think of some of the best marathoners in the world, Haile Gebrselassie and others like that, that's where they come from. But then when you think of the people who are winning sprinters, at least in the context of Africa, they almost all come from West Africa, from Ghana. And many people, when you start to look at the people who are on the podium or on the starting line of the final in the 100 meters in the Olympics, they can trace their lineage back to particular places, which, again, I think serves to emphasize that the genetic predisposition is really important. It's clearly going to be different. And it's quite interesting to think, I mean, way back, I'm going now, as I mentioned to you earlier, I'd started in exercise physiology, and I remember we had, there was a silver medalist who was a professor in our class, and he'd been a silver medalist in the 100 meters. And he had taught us that while power is really important in sprinting, he talked about the study where you could take sprinters and just ask them to do this on the table. To basically turn 2 fingers back and forward as fast as they can. And they'd studied the sprinters and found that they basically could do that faster than other people. So it wasn't just about power. It was about the ability to turn muscles that were agonist and antagonist at a given joint over really fast. So it was also neurologically encoded. So you can imagine that by taking a similar approach, as we've talked about here, taking the people at the extremes and sequencing their genomes and then really looking at them in detail, we might be able to find genes and variants that could direct us towards therapies for neurological disease, for joint and bone and muscle problems. Thank you.
The one question I have is firstly on the 5-day challenge.
Mm-hmm.
What was the nutritional element? What did they drink? What did they eat? Get an idea around it. So that's sort of one part of it.
Yeah.
Something totally different, or maybe related or unrelated, is talk about blood circulation as an important component of day-to-day health. And there are devices out there. One rate of weight. There are results out there that say that in health. So is this currently being used on athletes
Yeah. Okay. So 2 questions there. Let me deal with the first one first. So nutrition. The best answer to that which is, what do they eat? Is constantly. They eat constantly throughout this race. If you think about, you might have heard of the wall. That there's this idea that when you run a marathon, after like about, depending how fast you're going, an hour and a half, 2 hours, you reach a point that's much harder to get over, and a lot of people fall off at that point. The thought is that that's the point at which your glycogen stores, the stores of readily available glucose in your body, mostly in your liver, but also in your skeletal muscles, it runs out, and you have to start making the fuel that you're using in real time. Or you have to essentially be ingesting it in real time, or you'll rapidly fall off. So you can imagine, to go for 4 or 5 days, after the first few hours, 4 or 5 hours, if you aren't continuously ingesting fuel, you're just going to fall off. And the teams that win have a very, very, so my friend and colleague I mentioned, Mikhail, who's in the World Championship. So currently, I mean, they have, each one of them takes a role, and they have one person who deals with the nutrition for the whole team. Because they all actually have to keep up with each other. They're not allowed to be more than 100 feet apart during the whole race. And so there's an incredible science now of exactly how to do that. They're allowed to, they don't have to, there are some tournaments where they have to carry all their food. That's a huge challenge, of course. But there are most others that are allowed to drop off food supplies at various points, because it's a bit of an orienteering as well. You have to get to this point and get a marker. And then some of them have rest periods where they can actually refuel, But essentially, they eat constantly, and they try to find the highest, most dense, most calorific food possible. And they'll cook. You saw them cooking in the TV show too. So it's a huge challenge, and there's a whole science behind that. It's actually a whole other interesting talk that's probably someone else should give, but about the nutrition required to support this. And then you also asked about external counterpulsation. So this is an approach that has been used really for people with severe chest pain and coronary artery disease, to try and sort of encourage new blood vessel growth by squeezing the limbs kind of in a regular manner. And you sort of get strapped into the machine and your legs and arms, and it squeezes you, and you kind of, if you look at someone who's doing it, it looks like they're doing exercise, but they're actually passively, they're not moving. It's the machine that's moving for them. And it's basically pumping blood through blood vessels. My approach to that mostly is that if you can exercise, you should exercise, right? That's a simple answer, right? There has never been, and maybe there never will be, but there has never been any therapy for pretty much any condition that is as effective as exercise. So we have these statins, we now have these PCSK9 inhibitors for cholesterol. Essentially, if you want to cut your risk of heart disease, there is nothing better that you can do than exercise. In fact, people often say, well, I don't have time to exercise, but if you calculate, this is a number that's calculated from a huge study, 500,000 people. If you exercise for 1 minute, you buy yourself 5 minutes of extra life. That's how potent it is. And that's exercising at moderate intensity. If you exercise at higher intensity, you probably buy yourself 7, 8, 9 minutes of extra life. There is nothing that we have that is better for you. For every system, whether it's your heart, your lungs, it reduces the risk of multiple cancers. It reduces your risk of osteoporosis. It reduces back pain. It improves your psychology. I could keep going. There's nothing better than exercise. So my major answer to that is, if you can exercise, you should. It grows blood vessels in the same way that ECP does. The reason ECP came about is because those are for people who can't exercise. They have significant disease that they really can't exercise. For the rest of us, we just got to get out. Walking will do it. Running is fine. Cycling, swimming, anything. Just move. Stand up. Sometimes in my talks, I make people stand up and move around. I should probably have done that, because you've all been sitting now for 45 minutes, except me. Okay, you'll get to walk around afterwards. Yeah, maybe 1 and then 2. Is that okay? Yeah, thanks. Yeah.
I was just wondering, you talked about the one therapy. What other therapies?
Yeah. Well, so we're obviously very hopeful. I mean, I guess what I would say is that we're still at quite an early stage. We've almost completed phase 1, which is that we've sequenced the genomes of 250 of these people. To give you an idea, though, half of those samples we collected, and half of the samples have been collected over the last 25 years by a collaborator in Canada. So it's actually very time-consuming to do this. And then the technology to do it is only here. So that's one of the exciting things, is it's only in the last few years we've been able to afford to sequence this many genomes. We've also got a slightly larger group of about 500 or 600 people, where we've done not quite the whole sequencing, but we've done 1 million points along their genome to help us look. And we're really just looking for the first time. But every time we have a new gene, we can potentially target it. So right now, I would say that there aren't many examples. But if I'm I'm invited back next year, then I might be able to tell you about some more. We're particularly excited about this one that's in the Andean Highlanders. I think we want to look and see if we can target that. Each one will require a little bit of work, but the real advantage of studying humans is that you can go from human to human, essentially, is that you already know it's relevant for humans. You might have heard there's a lot of challenges in translating work that's been done in mice and rats to humans, and many drugs fail because of that difference. The advantage of doing it this way is we start with humans. We already know for sure this works in humans. If we can just mimic the genetic mutation, and maybe even one day like create the genetic mutation, but I think at the moment mimicking makes much more sense, then we can do that. So great. One last question here. Can you just wait for the mic? Just 1 second. Just here.
You mentioned that the adventure racers I'm wondering if that.
Yeah, that's a really good question. And it's something that people have studied quite a lot. We haven't carried out any of those studies ourselves, but we're very familiar with them because we deal with a lot of elite athletes. The immediate effects that I showed here with your heart fatiguing a little, and even you can measure in the bloodstream the same, and I didn't show you the data, but in that study we measured the same enzyme that we measured to tell if someone's having a heart attack, and measured it in these individuals, and it was slightly raised. Which people sort of jump on that and go, oh, that means it must be bad. In reality, I think we just don't understand that well. What we're measuring when someone has a heart attack is a very high level. But in reality, probably cells, not so much in the heart probably, but cells are turning over in the body all the time, and we're probably picking that up. So there was a lot of focus on that at the time. When people have gone to look specifically at the health of athletes who've performed at a high level, in general, we do not find a signal for long-term harm. And I think it's a complicated question because there might be some kind of a signal. You think of the football players and concussion, there might be some equivalent of that. But it's outweighed by the fact that people who start life as athletes tend to be tuned in, and maybe they're even genetically determined to feel really great following exercise, and so they have more of a sense that they want to keep doing it. But they do tend to exercise through the rest of their lives. And that's such a powerful signal. It's such a powerful positive effect on health that they tend to be healthier than everyone else in the population through the rest of their lives. So if there is an effect, some small effect of a negative, like tiring out their bodies or beating up their bodies by doing this really extreme exercise, it's small enough that we can't really find it in the big signal of the fact that they exercise long term. All right, well, that's all we have time for, but thank you so much for coming and being here today.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.